Gram staining is a technique used to differentiate bacterial species into two large groups, that is, gram-positive and gram-negative. It is always the first step in the preliminary identification of a bacterial organism and also a valuable diagnostic tool in both clinical and research settings. The requirements for this technique are clean, dry and grease-free slides, a nichrome wire loop, the gram staining kit which contains 1% crystal violet, grams iodine, absolute alcohol and 1% basic fuchsin or safranin, a 24-hour-old mixed culture suspension of gram-positive and gram-negative bacteria, and a compound microscope with an oil immersion objective. First, we are going to start by washing the glass slide to make it oil or grease free. Even though it looks clean, it still needs a good wash just before starting the experiment. This step is important for preparing a good smear. You can wash it with a regular detergent and a gentle flow of tap water. Make sure that all of the detergent particles are thoroughly washed out. Keep the slide for drying over a pad of filter papers or any other blotting paper. The next step is to clean the workspace with the help of a good disinfectant. This step is essential to retain aseptic conditions and avoid contamination. Now, name the slide with the culture provided to you. And then, mark the area on the slide where you want to make your smear. It allows us to identify the smear part on the slide. The next step is smear preparation, for which we are going to use a nichrome loop. Sterilize the loop portion of the nichrome loop on an oxidizing flame. Let it cool down completely while in between the burners. Now, take a loopful of the culture provided to you aseptically. Spread it onto the slide by means of circular motion. This step could be done in a best way outside the burners. Now the number of loopfuls to be taken depends on the density of the culture suspension. Here, as it is a light suspension, we are taking minimum 2 loopfuls for making a good smear. But if you are provided with a dense suspension, then a single loopful would be quite enough. This would get you a good feel through the microscope. So as in here, the loop's job is done. Sterilize it once and keep aside. Pass the slide 2 to 3 times lightly through the flame for heat fixing the smear. Place the slide on the staining rack and flood the smear with 1% crystal violet. Allow it to react for exactly 1 minute. Discard the excess and rinse with a gentle flow of water. Now, Flood the smear with grams iodine and let stand for 1 minute. Grams iodine is a mordant which complexes with the primary stain and fixes it in the cell wall of the bacteria. Discard the excess and add a few drops of decolorizer to the smear. It helps in washing off the primary stain and gram-negative organisms. Immediately, rinse it with water. Now, flood the smear with 1% basic fuchsin and allow it to react for 1 minute. You can also use safranin for counter staining. Discard the excess and rinse it well with water. Make sure that no stained blot remains behind on the slide. Air dry it over a pad of filter papers. After the slide has completely dried, place a single drop of oil over the smear and observe it under 100x power of the microscope. Lower the lens completely and then raise it slowly with the help of coarse and fine adjustments until you get the right focus. Take care that you don't lower the lens while observing through the microscope so as to prevent any damage to the slide. This is the observation found under a light microscope. Gram-positive organisms stain purple, for example, Staphylococcus aureus, and gram-negative bacteria stain pink, for example, Escherichia coli. Gram-positive bacteria contains higher peptidoglycan content in their cell wall. Hence, upon the addition of decolorizer, the cell wall pores get closed up due to dehydration, and this results in retention of the crystal violet iodine complexes. Therefore, gram-positive bacteria stain purple. Whereas in gram-negative bacteria, due to lesser peptidoglycan and higher lipid content, the primary stain is able to leach into the decolorizer solvent, leaving the bacteria unstained. These unstained bacteria are visualized with the help of a counter stain, which is basic fuchsin or safranin. Therefore, gram-negative bacteria stain pink.